In case of Escherichia coli, it is a symbiotic bacteria. And for sake of the energy, the source of energy generally is the glucose. Why it is a preferred substrate is the glucose? It will be broken down very easily in 10 steps in case of the glycolysis to yield energy. In absence of the glucose, if the lactose, a disaccharide, is present in case of a medium, if we provide this lactose in the medium, and the lactose enter into the cell, and there is a necessary to hydrolyze the lactose into glucose and galactose. So there must be an enzyme that is beta galactosidase. So this beta galactosidase is synthesized only in the presence of the lactose. So the gene it is expressed in presence of the lactose to hydrolyze the lactose into glucose and galactose. And these glucose and galactose which is used as a source of energy. If lactose is not present in the medium, so there is no necessary that the gene need not be expressed at all. So the gene expression, it is metabolic, environmental, physiological. If it is present in this environment, the gene is going to be expressed. If it required for the physiology, metabolic activities, in absence of the glucose, for sake of energy, it depends upon the lactose. So, if lactose enters into the cell, many beta galactosidase molecules are formed to hydrolyze the lactose into glucose and the galactose. Next. And the gene regulation is very coordinated. So, all the genes must be coordinated for expression of what it is a particular character. If there is a miscoordination between the genes, even the zygote develop into embryo develop into adult organism, is control and coordination between the different sets of the gene. If any sequence, any regulation of the gene, it is not in a proper way, it is going to lead to what it is abnormality. So, even the zygote develop into embryo into adult organism, it must be coordinated. Without coordination, there is no proper expression. So, gene expression, it is metabolic, environmental, as well as physiological. So, E. coli is a symbiotic bacterium. The common source of the energy, the preferred substrate is the glucose. In absence of the glucose only, it depends upon the lactose. For hydrolysis of the lactose, what is the required enzyme is beta galactosidase. So, gene is expressed by synthesizing particular protein, that is the beta galactosidase, to hydrolyze the lactose into glucose and galactose, which is used as a source of energy.